Hello guys, this is Dr. Possibility from Excel Academy. Kindly make sure that you subscribe to our YouTube channel and make sure that you register with Excel Academy. In this tutorial, I'm just going to explain the process of gluconeogenesis. So one big question we have to ask ourselves is, what does the body do when someone is starving? What happens to the body when someone is fasting? So we need to understand that glucose is the major source of energy in the human being. Glucose is the major source of energy in the human being. But there are times where someone starves for a day or for more than a day, and this glucose will be finished in the human being. As a result, the body would detect the low concentration of glucose, and it will now depend on the process of gluconeogenesis for it to have a source of glucose. So what is really gluconeogenesis? So gluconeogenesis is just the process that involves the synthesis of glucose from non-carbohydrate precursors. We all know that glucose comes from what? Carbohydrate. The breakdown of carbohydrate can lead to the formation of what? Glucose. Now, when you talk of gluconeogenesis, this is where the body uses a non-carbohydrate precursor to come up with what? With the glucose. In short, this process is just the reverse of glycolysis, whereby pyruvate is being converted back to what? To glucose. So when you talk of glucose, you need to understand that glucose tends to be depleted when someone is starving or when someone normally fasts beyond a day. So when a person starves beyond a day, you need to know that the levels of glucose will be low. In short, sometimes you even discover that the glucose will be finished. So for example, we need to understand one thing, that this is because the brain normally relies on glucose, which is 120 grams per day, as a source of energy. So for our brain to interpret the information, for our brain to work, it normally relies on what? On glucose. Now, in a case where glucose levels have reduced, in a case glucose level has finished, what happens next? So what normally happens is that pyruvate will be converted to what? To glucose. Pyruvate will be converted to glucose, and this process is simply known as what? Gluconeogenesis, which is involving the synthesis of glucose from a non-carbohydrate precursor. So, like I have said, gluconeogenesis is just the conversion of what? Of pyruvate to glucose, and we know that glucose is normally acting as a source of energy. So it's very important for us to understand when and why does the process of gluconeogenesis takes place. So the process of gluconeogenesis is taking place because the levels of glucose have become low. So when the levels of glucose are low in the body cells will detect that, and they'll tell the body to start making glucose from pyruvate. Now, there are so many molecules that can be converted to pyruvate. There are so many molecules that can be converted to pyruvate, and these molecules are known as what? Glucogenic. So any molecule that can be converted to pyruvate so that we can have glucose. It's known as what? It's considered to be glucogenic. So a glucogenic molecule is just a molecule that can be converted to pyruvate so that we can have what? Glucose. A very good example of a glucogenic molecule can be what? Can be lactate and the alanine. So lactate and alanine are what? Glucogenic. That simply means that lactate and alanine can easily be converted to what? To pyruvate. So take note of this. Your examiner would love you to know which molecules are glucogenic. Now, it is not only lactate and alanine which are glucogenic. We also need to understand that glycerol is also glucogenic. Glycerol is also glucogenic, such that glycerol can be converted to glycerol phosphate. Glycerol can be converted to glycerol phosphate, and then after that you need to understand that this glycerol phosphate will now be converted to dihydroacetone phosphate, and this dihydroacetone phosphate can simply be converted to what? Pyruvate. Now, one thing we need to understand is the enzyme which is helping in the conversion of glycerol to glycerophosphate is what? Is glycerokinase. So glycerokinase is just an enzyme which is playing a role in the conversion of glucose to glycerophosphate. And this process normally depends on ATP, which act as a source of energy. So this ATP would donate a phosphate group, which we can be able to see that side, and this phosphate group, when it is added to glycerol, this will lead to the formation of glycerophosphate. Okay, the other thing that we have to consider is the formation of dihydroacetone phosphate. So dihydroacetone phosphate, this is the process which is helped by an enzyme called glycerol phosphate dehydrogenase. So glycerol phosphate dehydrogenase will help in the conversion of glycerol phosphate to dihydroxyacetone phosphate. All right. So with that in mind, we need to understand that it's not only the enzymes which are playing a role, there is also the presence of NAD plus, which was acting as an acceptor in the process of glycolysis. This time around, this guy will be converted to that guy. Okay, so let's quickly move on and look at this same process. So gluconeogenesis, like I have stated, is just the process by which pyruvate can be converted to what? To glucose. Now we have this diagram here, we have that diagram there. So we have different enzymes. We have the enzymes in black and the enzymes in red. So this process from glucose to pyruvate, this process is what? Glycolysis. We need to understand that this whole process here, which has been described here, it is just the process of glycolysis. Now, within the process of glycolysis, we can take note of these enzymes in red. So there is that enzyme in red, that enzyme in red, that enzyme in red, and that enzyme in red. So the enzyme in red belong to the what? Gluconeogenic pathway. So these enzymes, they play a role in the process of what? Gluconeogenesis. So that's one of the things that we need to understand. Take note. within these enzymes which we found in the process of glycolysis, there are enzymes that play a role in what? In gluconeogenic pathway. So the enzymes that play a role in gluconeogenic pathway are one, this one, which is glucose 6 phosphatase, then fructose 1,6 bisphosphatase, then you have phosphoenolpyruvate carboxylase, and you have pyruvate carboxylase. Okay, so these four enzymes are playing a role in what? Gluconeogenic pathway. Take note of all of them, the four of them. Then these reactions, they normally overcome the high energy free of the irreversible reactions of glycolysis. Now, the enzymes in blue are held in common between the two pathways. Okay, so let's quickly move on and look at some of the enzymes that are involved in the irreversible glycolysis. Okay, so we need to understand that the irreversible glycolytic enzymes are: one, we need to look at the hexokinase; two, we need to look at phosphofructokinase; then we have pyruvate kinase. So these three enzymes are irreversible glycolytic enzyme. Irreversible glycolytic enzyme. So the irreversible glycolytic enzymes are the ones which I have just simply given here. One, hexokinase; two, phosphofructokinase; three, pyruvate kinase. So you might be asked to state which enzymes are irreversible. Then we have the enzymes of gluconeogenesis. In short, the enzymes that take place in the process of gluconeogenesis. So if you remember very well, the enzymes that are taking place in gluconeogenesis are the ones which I marked red. So you have this one, you have that one, you have that one, and lastly you have that one. So let me quickly look at these enzymes. The first enzyme you need to know is pyruvate carboxylase. So you need to understand that this takes part in gluconeogenesis. The other one is phosphoenolpyruvate carboxylase. Then you have fructose 1,6 bisphosphate. Then you have glucose 6 phosphate. So these four enzymes are taking part in what? In glucogenesis. Taking part in gluconeogenesis. So make sure that you are able to understand that. Now, let's quickly discuss these enzymes one by one. So we need to understand that the first enzymes we can talk about is the pyruvate carboxylase. The first enzyme we can talk about is what? Pyruvate carboxylase. So pyruvate carboxylase plays a role in the reaction between pyruvate, carbon dioxide, ATP, and hydrogen to produce what? Oxyacetate plus ADP. So you need to understand that one phosphate group will be lost on this guy to produce ATP, a phosphate group, and two hydrogen ion. So in short, you need to understand one thing, that this reaction does not take place on its own. Okay, this reaction does not take place on its own. In short, this reaction does not only depend on this enzyme. This reaction does not only depend on this enzyme. Instead, this enzyme would depend on a vitamin which is known as what? Biotin. So biotin is an enzyme which plays a role in the conversion or in the what? In this reaction. Biotin is just a vitamin which is playing a role in this reaction. Quite alright, this enzyme is doing its job, but it needs biotin for it to take place. And you need to understand that biotin is just acting as a what? A cofactor, because it is not an enzyme, okay? It is a vitamin. So biotin is just acting as a what? A cofactor. But the main enzymes is what? Pyruvate carboxylase. So pyruvate carboxylase is able to fix what? Carbon dioxide. In short, this guy is just playing a role in fixing carbon dioxide. Is playing a role in fixing what? Carbon dioxide. Okay, and you need to understand that enzymes which are capable of fixing carbon dioxide, they normally require a cofactor. In this case, the cofactor is just biotin. Now, the question is, where does this reaction takes place? So this reaction normally takes place in the what? In the mitochondrial matrix. This reaction normally takes place in the mitochondrial reactions. Now, let's quickly look at the structure. So this is the structure of biotin. This is the structure of biotin, which is acting as a cofactor in this reaction. And take note that there are no components of ATP in the structure of biotin. So this biotin is just working as a what? As a cofactor. Okay. Let's quickly move on and look at the other enzymes. So take note of where this reaction is taking place. This reaction is normally taking place in the mitochondrial matrix. So let's quickly look at the other enzyme, which is phosphoenolpyruvate carboxylase. Carboxylase, phosphoenolpyruvate carboxylase. So this enzyme is just playing a role in the reaction between oxoacetate and the GTP. So you need to understand that in the previous reaction, one product was the oxoacetate. So this guy here will react with that guy, and this reaction will be helped by the presence of the enzyme, which is the phosphoenolpyruvate carboxylase. So when this one react with that one, it's going to give us phosphoenolpyruvate plus GTP plus carbon dioxide. So these enzymes, phosphoenolpyruvate carboxylase, will just help in the reaction of oxoacetate and the GTP. Remember, our major product should be pyruvate, and this pyruvate has to be converted to glucose. So where does this reaction takes place? So the other reaction took place in the mitochondrial matrix, but this one normally takes place in the cytosol. So this reaction is taking place in the cytosol. Now, when phosphoenolpyruvate, which I can simply say PEP, okay, I can just simply abbreviate it with PEP. When this guy is synthesized, it can lead to this reaction. So this is the reaction which can be summed up by PEP when it is being synthesized. And if you can see, there is pyruvate plus ATP plus GTP plus water to give us PEP, ADP, GTP, a phosphate group, and a hydrogen ion. Okay, so one thing we need to understand is that pyruvate is carboxylated in the mitochondria by pyruvate carboxylase. So pyruvate carboxylase is playing a role in the carboxylation of what? Of pyruvate. So pyruvate is carboxylated in the mitochondria by pyruvate carboxylase. So pyruvate carboxylase is playing a role in such an activity. Now, one thing we need to understand is that oxyacetate can pass out of the mitochondria. The oxyacetate which we saw, which was being created that side, it cannot pass through the mitochondria. But oxyacetate is decarboxylated and phosphorylated in the cytosol. So that's where this guy oxyacetate is being done, and this is helped by the enzymes, which is what? Phosphoenolpyruvate carboxylase. Okay guys, so make sure that you understand this concept and make sure that you are able to register with Excel Academy. Okay, the other enzyme we can look at is fructose 1,6 bisphosphatase. So that's the other enzyme we can look at. So fructose 1,6 bisphosphatase is just playing a role in the conversion of fructose 1,6 bisphosphate plus water to give us fructose 6 phosphate and a phosphate group. So fructose 1,6 bisphosphatase is an allosteric enzyme and it normally regulates gluconeogenesis. So this enzyme plays a role in the what? In the regulation of the process of gluconeogenesis. So the process of gluconeogenesis is normally regulated by the enzyme fructose 1,6 bisphosphatase, and this type of enzyme is known as an allosteric enzyme. Now, this fructose 1,6 bisphosphate is playing a role in the conversion of fructose 1,6 bisphosphate to what? To fructose 6 phosphate. So this is just easily converted to glucose 6 phosphate. So this guy which has been created, this one which has been created, it will easily be converted. Remember in glucolysis, we had to experience whereby glucose 6 phosphate was converted to fructose 6 phosphate, and that was by isomerization, isn't it? So that simply means that because this can be converted to that, and that reaction was reversible, because this can be converted to that, that means that even that can easily be converted to that one. The next enzyme, which is glucose 6 phosphatase. So glucose 6 phosphatase will now work on this one. So first, fructose 1,6 biphosphate helps in the conversion of fructose 1,6 bisphosphate to fructose 6 phosphate, and this fructose 6 phosphate will easily be converted to glucose 6 phosphate, and that is by isomerization. Then glucose 6 phosphatase will react on glucose 6 phosphatase to produce what? Glucose, which is a major source of energy. Remember, our major goal was to have glucose. So when glucose 6 phosphatase catalyze the reaction between glucose 6-phosphate and water, it will give us what? It will give us glucose. So this reaction, it is just helping to remove what? To remove a phosphate group. To remove a phosphate group. So a phosphate group has to be removed on glucose, and this phosphate group is normally attached on carbon number 6. So a phosphate group is removed, and when the phosphate group is removed, it's going to give us what? Glucose and a phosphate group. Now after that, the liver will now send glucose to blood, and it's going to maintain the process of homeostasis. Because remember, the reason why this process took place was because of lack of glucose in the body. So lack of glucose in the body is the one which made the body to detect that there is no glucose. So when this glucose is being converted or it's being formed, you need to understand that the liver now will send the glucose to the what? To the blood, to the bloodstreams, and this blood will simply go to the cells, and this glucose in the cell will normally act as a source of energy. All right. Okay. Now you need to understand one thing. Glucose 6-phosphate. Glucose 6-phosphate is just a variable, and it is a precursor of what? Of glycogen synthesis. Okay. And then another thing which we need to understand is that glucose 6-phosphatase is present only in tissues responsible for maintaining blood glucose, such as the liver and the kidney. So this enzyme here can only be found in organs such as the liver and the kidney, and these two are just responsible to maintain the what? The blood glucose. And this process of maintaining the blood glucose, we are just simply talking about homeostasis. So the liver and the kidney are playing a role in the maintenance of blood glucose in the body. And as a result, you need to understand that because of that responsibility, they are one of the few organs that are able to have this enzyme. Okay. So guys, with this being said, please make sure that you subscribe to our YouTube channel, and make sure that you also contact us in case you want to register with that. Then one thing you need to understand is that this glucose 6-phosphates, it has to be regulated, and the organ which is responsible for the regulation is just the liver. So the liver is the one which normally regulates glucose 6-phosphatase. Remember this point, whereby we need to understand that glucose 6-phosphatase, it is playing a role in the conversion of glucose 6-phosphate to what? To glucose, and glucose will help us to have energy in the body. So the amount of glucose 6-phosphatase is regulated by the what? By the liver. Okay. So the liver, this is just showing what happens in the liver, whereby this is the liver, you have the cytosolic side, and then you have the lumen side here. So you need to understand that the glucose 6-phosphate will be converted to glucose and the phosphate group, and these guys will simply leave. Okay, and this guy, it will normally be an enzyme which will help in the conversion. So when this guy is being converted to that, these two, because they are the one which are responsible for energy. They are the ones which we want. They are going to be released outside the cell, and this is where you are going to find the what? The blood, and the blood will simply collect the glucose. Okay, so guys, please make sure that you make sure that you subscribe to our YouTube channel, and make sure that you also register with Excel Academy. So when you talk of the stoichiometry of gluconeogenesis, you need to understand that 2 molecules of pyruvate are used, 4 ATPs are used, and then 2 GTP are used, 2 NADH are used, 6 molecules are used to just produce 1 molecule of glucose, So for you to produce 1 molecule of glucose, you need 2 pyruvate molecule and 4 ATP. If you remember very well, in glycolysis, 2 molecules of pyruvate were being produced, and then 2 molecules of ATP were being produced. Okay, so we need to take note that it takes 6 nucleotide triphosphate molecules to synthesize glucose, and only 2 nucleotide triphosphate molecules are generated from glycolysis. And then it takes 4 extra high phosphoryl transfer potential molecules to drive the unfavorable gluconeogenesis pathway. Alright guys, so please make sure that you follow up my next tutorial, and make sure that you subscribe to my YouTube channel. So guys, this was Dr. Possibility from Excel Academy. Please make sure that you subscribe to our YouTube channel.